Indirect immunofluorescent antibody test. What is IFAT? Indirect immunofluorescent antibody test (IFAT) is a technique used in laboratories to detect circulating autoantibodies in patient serum. It is used to diagnose autoimmune blistering disease. For example, it is used to detect anti-nuclear antibodies in systematic lupus disease. This technique uses pathogen-specific monoclonal antivirus antibody as the primary antibody and fluorescent isothiocyanate conjugated antisera to the immunoglobulin used for the primary antibody.
So, what is the component of this assay? This assay contains certain markers. The markers for this indirect fluorescent antibody technique are composed of two components. First is the monoclonal antibody, and the second one is the fluorescent stain, or also known as the fluorochrome. For example, this method can be used to diagnose certain diseases such as syphilis and SLE. In syphilis, the key is to detect the anti-treponemal antibodies that bind to antigen T pallidum cells, while in systemic lupus erythematosus (SLE), this method were used to detect the presence of anti-nuclear antibody.
For direct immunofluorescence, the fluorochrome-labeled antibody binds directly to the antigen on tissue section. However, in indirect immunofluorescence, an unlabeled antibody binds to the antigen first. Then, a fluorochrome-labeled secondary antibody or anti-IG binds to it. Since this secondary antibody binds to the antigen indirectly, it is called indirect fluorescent antibody test or IFA test. Indirect method is actually more sensitive compared to the direct method because more than one fluorescent containing antibody can bind to the primary antibody.
Since the indirect immunofluorescent antibody test is a qualitative test, the results can be obtained by observing the slides under a microscope. Hence, a titer is not required. The positive control is important as it is crucial to determine whether something went wrong during the staining process. It can be any tissue or cells where the protein of interest is known to be expressed in abundance. Positive controls should display staining. This means that the focal cluster of cells should exhibit apple-green cytoplasmic fluorescence when observed.
For positive results of the indirect immunofluorescent antibody test, antibodies will be labeled with a fluorescent stain which gives a green-yellow color in ultraviolet ray. The sample will appear similar to the positive control at one of the time points. For negative controls results, there should be no fluorescence can be seen as there is no fixation reaction. Negative results means there is no significant difference in background fluorescence staining in positive and negative cells.
The diagram on the left-hand side displays a positive reaction of detection for immunoglobulin G antibody to Toxoplasma gondii in pregnant woman with a bright and greenish yellow color. On the other hand, the diagram on the right-hand side shows a negative result with non-fluorescent staining. For your information, Toxoplasma gondii is a protozoan parasite that infects most species of warm-blooded animals, including humans, and causes the disease toxoplasmosis.
Next, we will talk about the application of indirect IFAT in research purposes. Other than that, this technique also can be used to detect antibodies in aborted fetus. So this technique was used to titrate antibodies in bovine sera to parainfluenza 3, infectious bovine rhinotracheitis virus, and also bovine viral diarrhea virus. Finally, in this research, the presence and the absence of antibodies were determined from 100 aborted fetus and also 50 non-aborted.
Next, I will talk about the research of indirect immunofluorescence tests, which is conducted in Sweden. Sudden surge of dengue cases in Sweden caused researchers to find alternative serological tests for diagnosis of dengue fever. Luckily, they found out that indirect immunofluorescence test is another good alternative.
IFAT has been the most common serological test used for diagnosis of leishmaniasis, a disease caused by a group of protozoan parasites called leishmania, which affects poor countries like India, Bangladesh, and Nepal. According to the World Organization for Animal Health, the IFAT is one of the most suitable diagnostic tests for leishmaniasis. Despite the fact that IFAT is not a perfect test, it is frequently used as a reference test for the relative validation of new diagnostic methods and to estimate the true canine leishmaniasis prevalence.
That's all from us. Thank you.